If you just do it, it'll turn out okay. Hey guys, back to More Talking Time with Caffeine. You knew one of my guests from the last episode, Dan Creation Myths, but I also got a new guest today. Introduce yourself.
Hey everyone, I'm David Neff. I will be studying biology at the University of Maryland, Baltimore County in fall of 2021. I am super excited for this chat because ERVs are some of my absolute favorite subjects in molecular biology and genetics. It's so much fun. So I've been looking forward to this for quite some time now.
Oh yeah. So last time Dan was here, we talked about viruses and stuff and how they work and how to fight them and how they attack the body and attack cells. But what would happen if the virus stayed around and say we gave the virus to our kids and our grandkids?
Now we're just stuck with it, and you do that enough times and you end up with 8% of your genome being the leftovers of dead viruses, which is where we are right now. We are more dead virus than we are human protein encoding gene by a large amount because of these things called endogenous retroviruses, which is so darn cool.
So a virus decides to attack, say, the thing that makes sperm cells, or the sperm cells itself, or the egg cells itself, or
basically, yeah, it has to infect the germline, and then a descendant. So the germline within your body, the germline are the cells that make either egg or sperm, and then a virus has to get in there, and specifically a retrovirus, so a virus that integrates itself into your genome. The way retroviruses work, they're super weird. They're RNA rather than DNA. So our genome is DNA. Retroviruses have an RNA genome, but they do this thing called reverse transcription. They go from RNA back to DNA. And we call that retrotranscription because, like, normal transcription is DNA to RNA. That's what we do when we express genes. Retroviruses go the other way and then cut a little hole in your genome and add themselves, and they just stick them right in there. And when they do that in a germline cell, you could pass it on to your kids.
So like, unlike regular viruses, they didn't like attack the cell and destroy it. They build themselves in there like a little like piggyback or something.
They can do either. So they can. So probably the best known retrovirus is HIV, which very famously attacks the cells of the immune system. And so what it does is it will get in and it will do the normal virus thing of get in, take over, make more of itself, and then burst out and go attack other cells. But that's not the only thing it does. It also can get into cells and kind of go into stealth mode and just integrate into the genome and just hang out. And for that reason, that's the reason we can't cure an HIV infection right now, or at least not typically. There have been a few cases that maybe, but those are exceptions. What happens right now with the really good HIV drugs that we have is that you can prevent any viruses from being circulating in the system. But what you can't do is cut it out. So if a person who is HIV positive was to stop taking the medication, then those integrated viral genomes could, in theory, reactivate and then cause illness. So they're in there. They can do the normal virus thing, but they do this extra thing where they just hang out and transmit as part of the host DNA, which is weird and opens up a lot of interesting evolutionary dynamics that most viruses don't have.
Okay.
And what I particularly love most about ERVs is that we can construct a phylogeny, an evolutionary phylogeny based upon endogenous retroviruses, and it matches perfectly from what we would expect from other methods. So it acts as a independent confirmation for what we already know from other fields of study. And I am pulling something up right now that demonstrates just that.
Cool. And also ERVs, I guess they had to, I'll say lucky they have to hope that the person they affect is like is someone whose genes death spread with the rest of the community, or that virus's line is dead too.
It leads to some really interesting evolutionary dynamics, because once you integrate, you're now kind of at the mercy of your host. And if you kill your host, well, now you're not trained, you're done. So it's actually, what we actually expect to see is selection to minimize the damage done once the integration has occurred and you've kind of gotten stuck.
Yeah, so here's a phylogeny that I basically found based upon endogenous retroviruses. This is from the talk.origins website. 29 evidence from macroevolution, their study on endogenous retroviruses. Very interesting how we can actually do this. We see that humans and chimps are the closest relative, exactly what we already know from DNA sequencing, et cetera, So this is very powerful evidence for common ancestry.
All right.
Yeah, it's super cool. It's super cool. And to put some numbers on that, with humans and chimps, there are about, and I'm using approximate numbers here, but there are about 200,000 retroviral insertions in humans and chimps, and like in each one, and over 99% of them are shared between the two. You can look at a specific place in the human genome, find an insertion in ERV, endogenous retrovirus. You look at that exact same place in the chimp genome, you find the same viral insertion. It's just beautiful the way it shows the pattern of common ancestry. It's really cool.
So what are the YC people that say that it's not a good concept for evolution? The virus could attack the same animal twice, different animals twice.
That's wrong, because we can actually watch in real time how these viruses target the genome. So, like with HIV, for example, we can document in people with HIV where the insertion sites are, and what creationists will say is, well, they have specific sites that they insert at. That's not true. They have preferences, and I'm using preferences. It's not. They don't have desires. They preferentially will target certain parts of the genome. HIV, for example, tends to integrate in gene-rich parts of the genome. But if you look at where HIV integrates, it integrates all over the genome, many different sites spread out on every chromosome in the genome. There's no one or handful of places where HIV integrates. That's true for other retroviruses as well. So you can find what we call hot spots and cold spots, where insertions are more likely or less likely, but it's not deterministic. So if you find two endogenous retroviruses with the same sequence in the same place, the best conclusion from that is common ancestry. And when you find 100,000 of them or 200,000 of them in the same place, that's common ancestry. There's no way around that.
Like,
exactly. It's kind of, it'll be like at the end, if you gave your students a test and you had 500 students and only 2 people have the same exact error in the same exact location, you could probably conclude that one is copying from the other.
Something funky's going on there. Yep. Mm-hmm.
And I'm sure you've seen a lot of, and I'm sure as a professor, you've seen quite a bit of that.
Less than you might expect.
Yeah, well, that's probably because you're at Rutgers and not some local community college. But
I don't know. You get some of the worst cheating scandals at places like I won't name any names, but
yeah, true.
Ivy Leagues.
Let's take a look at some of the.
Real fast.
I'll go right ahead.
How far back can we trace ERVs? Can we trace it? Do we share any ERVs with our plant or fungus cousins that we know of?
Yeah. I don't know of any that are shared among like major eukaryotic groups, but within animals, I know you can find a bunch. I know certainly with tetrapods. Trying to think. Mammals definitely, because then you could find mice ones that are homologous. I'm not sure how far back you can go actually. They do degrade over time, so you can actually look and see, like, for example, all herbs have like a specific structure to them, where they have kind of end pieces, and then they have specific genes and stuff in the middle. And you can see that the ones like that humans have on our own are more complete than the ones we share with chimps, than the ones we share with chimps and gorillas, and so on back. The older they are, the more degraded they are. And most of the ones in our genome are just the end pieces. All the stuff in between has been degraded away. I'm not sure how far back you can go before that signal is gone, but I know that we can identify orthologous herbs within different mammals. I don't know beyond that off the top of my head.
I guess also since mutations can happen too in the virus, so they might change after they split after they split too, maybe.
Yeah. Mm-hmm. And once the changes happen in the virus that inactivate it, now you've removed the selective pressure to maintain it, right? Because if the virus is active, the ones that can propagate the most effectively are going to be maintained, and the ones that can't are not, because they're competing with each other. Once a virus is inactivated, well, now you can't reactivate out of the genome. You can't keep doing your thing. Well, that removes all the selective pressures to keep all those genes active. You inactivate one of them, and it removes the selection on all of them. So now you're stuck in the genome with nothing keeping you there, no selection to stay complete. So when the host, either through problems with replication or recombination, or there's some evidence that there's enzymatic targeting of these things by the host, because aha, virus, kill it, right? When those things happen, those losses accumulate because there's no selection to keep that virus there. So you would expect to lose that phylogenetic signal over time. I don't know how far back you can go and still detect it.
All right, so what are you going to say, David?
So I kind of found a couple of creationists' rebuttals to the ERVs, and oh boy, is it bad. Here's one of the actual arguments. Interpretation. However, we know that the evidence doesn't speak for itself. Part of the scientific method demands interpretation. Since every science has to interpret the evidence, their interpretation bias affects what they say about origins. So ERVs, in and of themselves, do not prove evolution any more than they prove creation. Since we know that the word of God is true, we know that ERVs cannot be proof of evolution. And they go on and say, ERVs are not junk, which is basically standing for truth's reply that they have function, therefore they're not evolution.
I actually did a little work on that one, and there are a couple of functional groups of herbs. Let me pull it up here, so I don't remember the numbers off the top of my head. But there are some functional groups of herbs, and I wish I could find it here. I have too many things open. What else is new, right? Hang on. I'll find it. Here we go. So there are some that are involved in regulating human gene expression. That makes sense because herbs contain promoters, and retroviruses often integrate in gene-rich regions. So that makes sense. We've co-opted several of those promoters. We know that they are used targeting for the organization of the genome within the nucleus. That makes sense because you've got these regions that are kind of not doing much else. So you could be like, aha, let's use this. And then the other one, and my favorite, is when we have actually co-opted the genes. And my favorite example of that is genes called syncytins that are used during the early development of the placenta. These are genes that have been acquired from retroviruses. They're not technically active herbs. They are genes that are under control of human promoters, but they were acquired from a retrovirus. And these genes make a protein that is basically a stealth mode protein. It suppresses an immune response. Or I should say, specifically, it suppresses the inflammatory response. And this is important because it allows the embryo to implant into the uterus more successfully. So you can compare between the marsupials and the eutherians, the true placental mammals, right? So we're eutherians. Marsupials make a rudimentary placenta, but their young are born very, very underdeveloped and have to finish developing in a pouch, think like a kangaroo. The reason for that is the uterus actually mounts an immune response to the implanting embryos and kicks them out relatively early in the process. But with the help of these retroviral genes, suppressing that inflammatory response, you're able to develop a much more robust placenta that allows for complete development in utero, in eutherians. And that wouldn't be the case if we didn't have these genes from the retroviruses. So, like, super cool.
Oh, another lame joke. Does that make Australia? Does this make Australia the origin of Pokemon, the original pocket monsters?
I get that joke. I get it. It took me a but I get pocket. I get it. I like that.
Oh. So
go ahead.
So on the picture that David shared earlier, I noticed there wasn't any ERVs in the New World monkeys. Do we don't know about them, or there was none?
Huh, that's a good question. I'm not sure. I'm going to Google that real fast because I honestly don't know. I believe I would think there are, but I don't know. Well, actually, I will say this. I don't know if there are any New World monkey specific herbs. There should be. But in that figure, I think there were ancestral ones that it would imply that they would be in the New World monkeys, but I don't let's see. New World. I'm going to just Google New World monkey exclusive herbs. Let's see what we find. I don't know.
I think that was that chart was just saying our. I guess that chart they were showing was just showing what we share of other the ones we share of other primates or other monkey, catarrhine, river, however you pronounce it.
Let's see. Yeah, it looks like they have them. Let's see. Genomes of all so all vertebrates have herbs. So that's first thing I just found. This is a paper from 2018. All vertebrates have herbs. So that answers that question. Let's see.
Sorry, chordates, you're out of luck.
Basal chordates. I mean, you would think, right? But I want to find a phylogeny of herbs among the Oh yeah, it looks like let's see, there are the primates, there are the lemurs. Yeah, it looks like there's herbs in all of them. Yeah, I mean, yeah, I'm sure they have their. I mean, they have to have their exclusive herbs. I don't know anything about New World monkey herbs, but they have to.
Yeah, that's totally why they came to the New World, escape from new viruses. That's why they came to get rid of we're not getting new viruses. Let's go escape to South America.
Right. Yeah, it's so let's see. So by the way, here's just so everyone's on the same page. The way we know these are viral inserts is very cool because retroviral genomes are super unique. They have 3 basic kind of components, and each component contains a number of different genes, but the components are GAG, G-A-G, POL, P-O-L, and ENV, E-N-V. Right? GAG, POL, and ENV. And each of those has multiple genes in it. And there's also a promoter in there. And the genetic structure is weird, and you express a polyprotein and then cut it. It's weird. Doesn't matter. They've got these three regions, right? And then when they insert, you also get LTRs, long tandem repeats on either side. So you get these LTRs with the genes in between. And then they do something really distinctive when they insert. When they land in your genome, wherever they cut, they make a copy of that. So if you have like a gene and then like a specific sequence, and the one I just happen to know there's an example in chimps where the kind of duplicated I think it's ATTAT. It doesn't matter what it is. Just give you an example. ATTAT. And then in humans, it's like gene, ATTAT, or region, whatever it is. You've got regions on either side that we can match to the chimp genome, and then we've got ATTAT in the middle. In the chimps, you've got region 1, ATTAT, LTR, GAGPOL-EMV, other LTR, ATTAT again, and then the region that matches to the human chromosome, right? So you've got these two regions in humans that are right next to each other. In the chimps, they're split apart. The region in between, that little bit is repeated on each side, and then you've got that retroviral structure in between. And that's like textbook retroviral insertion that's unique to the chimp lineage that's absent in the human lineage. And it's like you find those structures, whether it's the whole thing, some combination of those parts. Most of them are super degraded, and it's just the LTRs. Like, you find any of that stuff, that's a retroviral insertion. It's like, we know what these look like. We can watch them happen with things like HIV and other retroviruses that are circulating right now. We know what the insertions look like. So do these come from viruses is not an open question. Herbs come from viruses. They are the remnants of dead viruses that now make up 8% of the human genome. It's so cool that we could find that.
Hey, do we know if there's any viruses, different viruses, like within the human population that some humans have that others don't? Like
viruses
like
that humans have?
Like when we spread out out of Africa in different locations.
Yes. There is, I think there's one herb that is variable among humans. So some human lineage, I forget where geographically and what lineage have what, but if you survey all of humanity, there's one herb that some lineages have and some don't, and there's variation in the copy number. So a lot of times these things, when they mobilize, they'll cut themselves out. Instead of just spreading throughout the body, they'll just reinsert themselves somewhere else in the genome. So you get one copy over here, it copies itself out, but then just pastes itself down over here. So you get multiple copies in the genome. With this one particular herb, you get variations in copy number, and some human lineages don't have it, which tells you that this is something that has been mobilized. It's been active in terms of copying itself and cutting out and inserting somewhere else, like since human diversity like split from our most recent common ancestor, which is super cool. Now we're talking within the last 200,000 to 300,000 years, these viruses were actually active, which is so darn cool that we can see that.
Probably the same thing. Like we've at least traced our Denovian and Neanderthal cousins' DNA too. Do they have viruses that we don't have too?
That's a great question. I'm sure they do. I would expect them to. It was probably fewer than separate us from chimps, because you're talking between a few 100,000 years and a couple of million years for other members of genus Homo versus like on the low end 6 million years, on the high end 12 million years for the chimp lineage.
Yeah,
so I'm sure there are. Yeah.
At least the viruses that we didn't like breed back into us after we split.
Sorry, say that again.
At least the viruses that we didn't breed back into us after we split.
Right. Yeah. I mean, it could. Yeah, if it was in the germline, you could potentially see that happening. And I don't know. I don't think there are any associated with the Neanderthal markers that we've identified, but I can't say there aren't for certain. I don't know of any, but I can't say there aren't. Be an interesting question. I don't know. Something to look at.
Any input from you? Look, David, you there?
Yeah, I'm there.
Oh.
Trying to think. So yeah, we can actually look at the human ERVs as well and use that as a phylogeny and see, okay, can we trace human origins back just by ERVs alone? And what do you know? It matches up perfectly with what we'd expect.
Can we actually do that? That's super cool. I didn't realize we get that tight resolution on there.
I'm not sure how precise it is, but I'm pretty sure I saw something similar to that. See if I can find a paper real quick.
While he's looking, like the ERV, get into our sperm and egg DNA, do they replace the ligand? Do they replace in there, or they just insert themselves?
They insert themselves, and it's actually not just ERVs, but also retrotransposons and DNA transposons. These are all classes of what people broadly call mobile genetic elements. It's things that basically cut and add themselves, and then they can make a copy and go over here and add themselves over here. And the transmission of these things is thought to be one of the major mechanisms that drives the increase in size of genomes, because it's kind of a ratchet where once it's in, unless you have a targeted mechanism to clip it out, it might degrade over time, but I mean, it's there. And so it inserts, it adds itself. It's going to, it's a copy and paste. It's not replacing stuff. It's inserting. And over time, you end up with a huge portion of the genome that's these things. So ERVs are about 8% of the genome. And you've got other transposons, DNA and RNA, that are even bigger than that in terms of percentages. I think altogether, they make up something in the neighborhood of 45% of the human genome are these transposable elements, most of which are now inactivated. They've mutated and they're now stuck. They can't mobilize anymore, but the remnants are still there, and we can tell what it is based on the sequence. So you get this kind of ratchet effect where they integrate, they add to the genome, they'll mutate over time. Many will be inactivated. Some will acquire functions in that new position in the genome. But most just kind of sit there and hang out. And I'll say that many are actually targeted for inactivation, and it's thought that is part of the immune system, where if everybody's familiar with the concept of methylation, if you add methyl groups to the DNA, it inactivates it. And one thing that we think is kind of underlying methylation patterns across the genome is that we have these things like transposons and retroviruses that we don't want to be active, because they'll mess with your cells if they're at the very least they're taking resources.
Yeah, especially since when you pass them to their kids, when you pass them to our kids, they're in every single cell that kid has.
Yeah. Right. So it's beneficial to have a mechanism that shuts them down. So we have enzymes that go around and methylate the crap out of these areas. And you'll see creationists that'll say, well, that's evidence that it's functional because, it's specifically targeted for these things. And I see that the other way around. If you're methylating something and just causing it to condense so it can't be expressed at all, that's really strong evidence not only that is not functional, but that if you allowed it to mobilize, that may actually cause a problem.
Yeah. So once it passes on to the kid, is the virus dead in our descendants, or can it be reactivated once it's been passed on?
It depends. So most of them are like dead as a doornail because they've mutated and they've lost stuff, right? So you need an enzyme that's able to cut it out. You need an enzyme, you need a sequence to say, copy me, right? And then you need the reverse transcriptase to be able to do its replication. So if it loses any of those components, then it's dead. It can't do anything. But there are ERVs that are like complete enough that it could mobilize, and we do actually see some evidence of this where when there are problems with DNA methylation and like targeting retroviral sequences with methylation and other forms of inactivation, we do see increased transcription from these areas, and that's actually associated with various forms of cancer. So it's possible that we do. I think it's likely that most of them are like dead, and of the ones that are not, we do a really good job with our immune system of shutting them down. Right, we target those regions to make sure they're not messing around. But sometimes there's a problem, and they do mobilize, and there's evidence that is associated with various diseases. But that's a small fraction. There's
Yeah.
relatively few that are complete enough to be able to cause a problem.
Hey, I was thinking tracing viruses and stuff, and what Dave's still looking. Do we know of any? I know viruses mutate, so it's hard to, like, do you have any ERVs in our bodies that match with viruses that we know of nowadays and out there?
I don't know of any endogenous retroviruses that are really good sequence matches to circulating retroviruses. But I just don't know enough about it to be able to tell you yes or no. I know I think there are 3 classes of endogenous retroviruses, and if I had to guess, they probably have like common ancestry with a specific group of retroviruses, but
Okay.
I don't know enough about the specifics of them to be able to tell you.
I was wondering,
I was
wondering, since we could, I was just wondering because we could trace back that chloroplast match cyanobacteria and our mitochondria match the whatever bacteria they match.
Right. Yeah.
I just wonder.
Could we do that? Well, I'm looking it up because I wonder if we can. Yeah, there are 3 classes, and actually, it looks like they are. Just gonna look at. Let's see. I don't know if they. Okay.
So, like you, David, if you found.
I don't know. I don't know off the top of my head if they're similar enough to circulating viruses to be able to say they came from this group of retroviruses. I wouldn't be surprised, but I honestly wouldn't be surprised either way.
Okay.
Because they mutate retroviruses mutate relatively fast. So think about like HIV. It's hard to we don't have a vaccine against HIV because it one of the reasons anyway is that it has a relatively fast mutation rate. So even within a single person, you have enough variation within that population that a vaccine may only be able to target a small fraction of it. Given that means your phylogenetic signal is going to get pretty messy pretty fast.
And viruses, especially HIV, is perfect case against genetic entropy, the one of the worst creationist arguments in the book, because they would be extinct by now, given how fast they mutate.
It's true.
It's
true.
You remember last time you were here, we talked about bacterial phages and stuff.
Mm-hmm.
Right. I heard there's some viruses that attack other viruses.
There are. Yeah, they're called virophages, and they are relatively small viruses. And they basically parasitize enormous viruses. In a group of viruses, I'm going to say the whole name because it's fun. It's the NCLDVs, which stands for nucleocytoplasmic large DNA viruses, which is a mouthful, but it tells you exactly what they do. These are the big viruses. These are Mimi virus, and Mimi virus was the first one we found. It was the biggest virus for a little bit. Now it's like, I don't even think it's on the top 10 anymore. There's just tons of diversity in this group. One of them is Pandora virus. Another one I think is Medusa virus. Now we're naming them after Greek mythological characters, which is fantastic to me.
Yeah.
And these viruses infect amoeba, and they basically just take over the whole inside of the cell and make a big structure called the viral factory. So it's the Mimi virus factory or the Medusa virus factory or whatever. And these little viruses, these virophages, they can infect the amoeba as well. When they infect on their own, they don't do anything. When they infect at the same time as one of the big viruses, so you get the little virus and the big virus in the same cell, that Mimi virus factory, instead of churning out more Mimi viruses, it gets hijacked and turns out the little viruses instead. It's so cool. So it's a virus of a virus. It's called a virophage.
So too bad we can't find a viral phage for the HIV virus.
Yeah, right. You need to be really complex to have a virus that infects you, and HIV only has a handful of genes. Like I want to say 10-ish, but someone can correct me on that. Whereas these Mimi viruses, these giant viruses, have over 1,000. So they basically do everything for themselves, including a lot of things that viruses aren't supposed to do for themselves. The big viruses do for themselves. So a little virus can take advantage of that. They're opening themselves up to being parasitized by being so complex. Whereas smaller viruses that have fewer genes, there's nothing to parasitize.
Yeah, so it's like a big food, almost food chain.
It's so cool.
The bacteria attack the viruses, attack the bacteria. The other viruses attack the virus, attack the bacteria.
Yeah, and you could actually see this in Antarctica. I think it was. Was it Antarctica? It was somewhere there was. I forget if it was Antarctica or somewhere else. I want to say it was in Antarctica though, where they found there's a lake, it had algae, and the algae had a big algal bloom, and then the algal bloom all got infected by one of these giant viruses, and it killed most of the algae. But in the middle of this outbreak, where the algae are getting destroyed, all of a sudden the viruses, the big viruses, start getting killed because the outbreak of the giant viruses led to an outbreak of the virophages that killed all the giant viruses, and it let the algae come back up. So you had this circulating rocks, paper, scissors game, basically, of algae, giant virus, virophage within this lake. It's very cool dynamic, very kind of multi-tiered, complex ecological system. Kind of oh, David, was that you?
Yeah, sorry, I was echoing in the background. I was trying to pull up the video in the tab so I can make a quick comment here.
So you found some new information for us?
No, I was responding to the person in the sideboard chat.
Oh, what happened here?
Oh,
okay. This person has to be a Poe. There's no way.
Yeah,
There's
I know.
No way.
Yeah, he's been
actually this actually does
no
lead,
way.
This actually does to a real question, and that is at the beginning of the COVID pandemic, there were some conspiracy theories about COVID being lab made. If it actually was lab made, if there were a virus that was lab made, what would we actually expect, if that's even possible at all?
I mean, it's certainly possible. I mean, there's viruses as bioweapons is a strategy that goes back to, like, I mean, we didn't know what we were doing, but I mean, you can go back thousands of years and find like the stories of like catapulting, smallpox infected bodies over a city wall or something during a siege. But in the modern world, that looks like trying to take a pathogen of some kind and optimizing it for both transmission and mortality, which is not an easy thing to do. The US has a bioweapons, or had a bioweapons program. I'm sure we still have all the stuff, although I don't off the top of my head know how active it is. I'd assume it's still active. Lots of places have bioweapons programs. But with SARS coronavirus 2, you can do the phylogenetics on this virus. It falls out. You do a big phylogeny of coronaviruses, and you get one clade that's all the bat coronaviruses, and then right in there, nested within all the bat coronaviruses, is SARS coronavirus 2. It's not hard. If it was a bioweapon, it wouldn't look like that. It would be different.
Yeah.
Right.
So, speaking for like that, are either of you eligible for the vaccine yet?
I am not, unfortunately, but I hope to be soon.
Okay.
I am already vaccinated because I was actually in the Pfizer clinical trial, and I did not get the placebo. I got the real deal.
Awesome.
So I got that. I was unblinded a couple months ago, and I'm gonna print that letter out and stick it to my fridge because heck yeah.
Cool. Yeah. I heard they're going in the 50s and up pretty soon, so I'm getting closer. I'm the 40 and up range.
Alaska just opened it up to everybody over 16. Just today.
Time to move to Alaska.
That will be more of that soon.
Yeah, I heard.
The supply is greatly increasing.
I heard in New York they had to start IDing people because people out from more people were getting the virus that were even live in New York. That
doesn't surprise me at all. Yeah, I mean, at this point, we're getting to where we're going to have such a surplus very soon, within a couple of weeks, that it's going to be opened up to everybody relatively soon.
Yeah, and hopefully Biden actually is able to get his goal of millions of vaccines in his first 100 days. And if we're
already over 90 million.
Yeah, if we actually do what the CD says and everyone gets vaccinated, we can have this thing beat by the end of summer.
Yeah, easily. Yep. Oh, easily.
I wonder if the COVID virus attacked anyone's germ cells.
It does not integrate. It is not a retrovirus, so we don't have to worry about it. It is an RNA virus, but it's not retrotranscribing, so we don't have to worry about that.
Okay.
One thing we don't have to worry about: persistent infections, which is good.
Yeah, and I'm also glad, at least that they're aware of. I might have to let them that it's not one of those mosquito viruses either.
It is not a vectored virus. Yes, thankfully. That would be, that's a whole other thing. But on the upside, that actually opens up other avenues for attacking the virus. Because, like, if you kill the mosquitoes, no more virus. So that kind of goes both ways, right? So, like, malaria is not a virus, but it's a mosquito vectored parasite. And going after malaria is hard because it's resistant to most of the drugs now. Or at least most malaria instances are at least partially resistant to various treatments. Many still work, but it's dicier than it was, say, 40 years ago. But if you can kill the mosquitoes, and we have things like CRISPR and gene drives that we're gonna start doing that soon, and I'm really excited. I am fully in favor of killing all the mosquitoes. Kill all the mosquitoes.
Yep, I agree. They're evil.
Yeah. This guy, I see him in other videos too, like Dapper Dinos and Erica's videos. His propaganda, this chat doesn't nothing to do with the subject.
I mean, can I block him?
You want to?
Yeah.
We're gonna use a timeout.
Yeah.
And then we'll see what happens.
Yeah. I first saw him in one of my videos about. I was doing a video about not even about science. It was about fan fiction, like a video game fan fiction. I was interviewing this guy who wrote these stories about these video game characters, and here can he come talking about flat Earth and evolution is wrong. I'm like, good for your stupid opinion, but that doesn't do the stuff we're talking about.
Yeah, not even the venue. I draw the line at like COVID trutherism and propaganda. Like,
same here.
If you're anti-vaxxing, I'm goodbye, delete.
Yeah,
see you later. You're blocked.
I will say this: anti-vaxxers are murderers. Point blank. If you're spreading deadly disinformation about vaccines, you're a murderer. Plain and simple.
I guess there's 3 levels of stupidity. Stupid. There's the YC people, then there's the flat earthers, and then there's the anti-vaxxers. Top of the list. And usually they're all the same. Usually they're all the same people. Usually some are other ones, but usually the anti-vaxxers are also like Kenny here is also a flat earther, and if you're that far in there, you're probably all 3.
The thing so people actually often ask, like, Dan, why do you bother with creationists? What harm does it do? They're going to say that the world is sick. That who cares? Real scientists don't care, right? Like, and that's true. The problem, as I see it, is exactly this, because if you can train your brain to believe something that is clearly not true, that is in conflict with all of the available evidence we have, if you train yourself to believe that thing anyway, then you could just as easily train yourself to believe something that's harmful, like anti-vax, as you can something that's relatively harmless, like young Earth, right?
Yeah.
It's the same mental exercise, the not just losing the ability to critically evaluate information, but practicing undoing those skills and being actively able to reject good information and actively unable to reject bad information. Like, it's the same set of skills, and if you train your brain to do it for one thing that's silly and harmless, then that can have harmful consequences down the road, and that's the problem.
Yeah.
And at the same time, you can cut that off. If you can train, you can teach critical thinking, and you can, like, how to evaluate data and how to actually, like, evaluate scientific hypotheses. You can apply that to something that is not controversial. Those skills carry over to other things, whether they are harmful or not.
Yeah, I used to say this a little bit. Sometimes I give the YECers sometimes that aren't like doing this on purpose, for money, the benefit of the doubt. Sometimes because they've only had to accept that evolution is a thing for like maybe like 100, 200 years now. Flat Earthers had to accept the deal like almost 2,000 years at this point.
Flat, yeah. I got nothing for flat Earth. We literally have pictures of the planet of Earth taken from outer space. We literally have pictures. I got nothing.
They've been photoshopped.
I mean, I can look at the moon and see the curved shadow on the moon.
The thing of
it's curved, guys. It's not
exactly.
a line.
Yeah, the thing with young Earth creationism is that it's a special kind of pseudoscience that it is refuted by literally every single fact of science. Like, there is not a single fact in any field of science that gives even an iota of credence to young Earth creationism.
Yeah, I thought it was interesting, not the evolutionary thing, but abiogenesis and stuff. We're going all over the world, we're jumping all over the place in these topics, but that you can that some of them might have formed in space through meteors and stuff.
Yeah, and we found like organic stuff like on meteorites, so it's one of the hypotheses, and it's not impossible. Yeah.
So have we found viruses on meteorites?
No, we haven't found anything more complex than very simple organic compounds. Viruses, you need something to help them, replicate, because they can't do it on their own. So you're only going to find viruses, at least as far as we know, in an environment where there are also cells. You're not going to find viruses outside of that, because they need something to replicate them. And even the biggest, most complex viruses, there's a few things that they can't do on their own. But I will say that it gets a little bit weird, because the definition of virus is not as simple as you might think. The simplest definition of virus is a intracellular parasite that transmits via, an extracellular capsid enclosed stage. But we can find plasmids that we've actually watched them in the lab adapt to transmit via little bubbles of fat called liposomes. And that's pretty darn close to a virus. It's like, well, what is that thing? It's a plasmid. We know what it is. It's a plasmid. It's a little circular piece of DNA you find in cells, mostly in prokaryotes, but not exclusively. Go ahead, Nev.
Yeah, I pretty much don't really have much else to say.
Yeah, so these plasmids, we know what they are. We can and then in this one experiment in the lab, they found the plasmids like going between, you got a cell, you got another cell, and you got a plasmid that'll be like, I'm in here, and then it'll make a copy of itself and kind of blob off and move over to this other cell. It's like, you're not supposed to be able to do that. How did you do that? That's how viruses work, and you're not a virus, you're a plasmid. What just happened? And now we kind of the definition gets a little bit weird once you start getting into stuff like that. So,
yeah,
as in all things in biology, there are no hard and fast lines. There's just a wide swath of gray.
Yeah,
and that's where all the fun is.
So it's like it's probably hard to, if at all, the trace viruses through our phylogeny tree. If they're separate or connected to them, or whatever.
Yeah, right. Once it's part of our genome, is it a separate entity? And is it undergoing like a kind of selection on its own? And you see some really interesting dynamics. So once it integrates and it gets stuck, you would expect to see selection not just to not hurt us, because it only reproduces when we reproduce, right? You expect to see selection to help us. And you actually do see this in some bacteriophages. So there are bacteriophages that infect cyanobacteria. And those bacteriophages, they can integrate into the genome. I think they're temperate phages that can integrate. But these phages have with them genes to increase the rate of photosynthesis. So when they infect a cell, that cell generates more energy for itself. basically giving the virus-infected cell a competitive advantage against the other cells in the population.
So if they can attack the cyanobacteria, can they also attack ones that are already chloroplast, or is that they protected inside the eukaryotic?
I don't think so, because I don't think they can get into the eukaryotic cells. I think they're limited to getting into the bacterial cells, and they can't get across the eukaryotic membranes because biochemically they're going to be very different. Yeah. Although that would be really interesting, and I'm trying to think now. I am not aware of any viruses that infect plastids. But goodness, I wouldn't, now that you mention it, I wouldn't be surprised at all. And I actually know who to ask about that too. So that's an interesting question.
Nice.
Yeah, there's a whole subfield called cellular metagenomics. So metagenomics is when you just like survey from the environment and just like, what DNA do you find? Just sequence everything and just see what you find. Cellular metagenomics or single cellular meta is when you take a single like an amoeba or an alga, right? And just within that one cell, sequence everything. And you find viruses and parasitic bacteria. You get your plastid DNA. You find all kinds of weird stuff in there. So I wonder if there's any phage in there that infects the plastids. That's a really interesting idea. And I've literally never thought of that until right now. But that is something I'm going to want to look into.
I started a new conversation out there now.
There we go. I want to look at plastid. I don't know what you call that thing. I guess you would call it a phage, right?
When a virus does, when a retrovirus does insert itself to a cell part of DNA, is that the same as endosymbiosis?
So a virus is different from endosymbiosis. Endosymbiosis, like the process, is when a big cell engulfs a little cell, and then that little cell, rather than getting digested, it over generations becomes a component of the larger cell. So you have predators that were eating cyanobacteria, and over time, those cyanobacteria, instead of breaking down, they became chloroplasts and other plastids. And so when a virus gets in, instead of and you could think of, I should say, we think of endosymbiosis, or we learned about endosymbiosis as like a partnership, but it's not that. It is a fight to the death, where the big cell is like, I'm going to eat you, and the little cell is like, no, you're not. And they're fighting within the big cell. And the equilibrium they arrive at is an organelle, right? A chloroplast, mitochondria, or whatever. With viruses, because when they get in, they break down. The first thing that happens is you lose the structure, right? The virus doesn't stay intact. So you don't have that same opportunity. So when viruses become part of the host cell, the way they do that is by integrating into the genome rather than staying as a discrete structure like you would see in endosymbiosis. Because when a virus gets in, you open up that capsid, you release the genome, like the viral particle that infects a cell then ceases to exist. It breaks down so the virus can do its thing. So there's no physical entity to become part of that host cell. It's just the DNA or the RNA is what matters. And then that happens all the time, right? That's 8% of our genome are those things that stuck around.
And some viruses are just RNA viruses, others are DNA viruses.
Yeah, they're everything. So you've got double-stranded DNA viruses. That's normal, right? We use double-stranded DNA. That's reasonable. Single-stranded DNA viruses. That's a little weird. Double-stranded RNA viruses. Also weird. We tend to think of RNA as single-stranded. You've got both positive and negative sense single-stranded RNA, which means is the genome mRNA directly, or is it complementary to mRNA? So you've got two different flavors of single-stranded single-stranded RNA viruses. Then you've got the retrotranscribing viruses, which I believe there are two groups of those. There's the canonical retroviruses, and then there's another group that does retrotranscription, and I forget exactly what those are called. But for anyone watching, there's a system of viral classification based on the genome structure. It's called the Baltimore system. And it's class 1, class 2, class 3, class 4, and it's based on the different genomic architectures. Yeah, and the fun part is that all these different groups and even different lineages within the different groups have independent origins. So viruses are massively polyphyletic. We're talking dozens of different origins, leading to all these different genomic architectures, which is super cool.
right? And if young Earth creationism was correct and everything was in distinct, neat kinds, then that's what we should see.
Yeah, that's one of my favorite tests of universal common ancestry for cells, because cells all use double-stranded DNA and use it the exact same way, whereas viruses, which we know do not share common ancestry, what does that look like? Well, they have all different genomes. Like their genome structures are all different. Cells all have the same genome structure. Right away, that's really strong evidence that they're common ancestry.
Yeah. I think I mentioned this in the last episode about viruses that I always wondered if maybe if we had a common ancestor with viruses that split off, the viruses went one direction and the cells went another direction when we were still just doing our enabled hypothesis.
I think some viruses do. I think there are some groups of viruses that share common ancestry with cells, and others definitely don't. Like there's a group of viruses that infect plants that like we know they evolved from a specific group of plasmids. And like we could just do the phylogeny and you get this phylogeny of plasmids. And then in the middle of this, there's this one branch of viruses all on their own that are unconnected to anything else. Like, okay, well, we know where they came from. There's some people that'll argue that those big viruses I mentioned earlier ought to be like a fourth domain of life. So you've got your bacteria, archaea, and eukarya, and then you should have the viruses sticking off there. It's a whole big viral phylogenetics is just a big, glorious mess, and we don't have answers for a lot of it.
They probably mutate faster. Viruses probably mutate faster than the cells do.
Yeah. So you lose the phylogenetic signal really fast. So like, even if there is common ancestry between some groups, after a couple 100,000 years, you're not going to be able to detect it anyway, because you get what's called saturation, where any two nucleotides in these two genomes that's the same, the probability of being the same through convergence is equal to the probability of similarity due to common ancestry. And at that point, it no longer becomes informative in terms of their evolutionary histories.
Yeah.
So you run up against that problem.
You said convergence. Is that like some YCs say that the virus could attack the same place, different location?
When we say converge, we mean like it's like when you work backwards in the past, like how you have two different sequences, how given the mutation rates, how far in the past would they have had a common ancestor? And the problems for viruses are that they have such small genomes and they mutate so fast that, like for humans, it's easy to like track that signal back because we get a big genome and slow mutation rates. For viruses with a tiny genome and fast mutation rates, that signal gets real muddy as you go back, even in the order of thousands of years.
Okay, I thought you should think like conversion evolution, like with dolphins and ichthyosaurs.
Oh, I see what you're saying. Yeah, exactly. So you could have different groups of viruses where you might have like a similar gene that ends up looking the same, not due to common ancestry, but it looks really similar because they mutate so fast and there's just not that many differences. Like there's not that many sites to mess with. So you can end up at convergence just by chance. And then your phylogeny is all messed up. It's what makes it's part of the charm of viruses. Is it's hard to figure out what happened.
They're clever little girls. Those viruses.
They're great. It's what makes it really is. Like you talk about like the origins of cells and go, okay, they had this, we kind of figure it out. The origins of viruses, and it's a huge mess. And it's great. It's so much fun.
Yeah, we may talk about animal and vertebrate viruses because that's us, and we like stuff that's like us. But is there people that study ERVs of like plants, how far they go back? They start plant ERVs. I'm
sure there are people that do that. I don't know at all of that field. My to the extent I have any expertise in endogenous retroviruses, it's purely among animals. I don't know about them in any other context beyond that. I'd be astonished if they didn't exist. But I don't know. I don't know for sure.
Like say if we can see a virus common between, like, say a daisy and an apple tree or something, or a seaweed, or I forget how plant phylogenies work.
Yeah, you would expect to find it like, so the four main groups, you've got your non-vascular plants like your mosses, and then you got your seedless vascular plants like your ferns, and then your gymnosperms, which are
Like
seed plants, but no flowers,
like pine
and they're
trees.
angiosperms. And that's all. I'm trying to think. Plants, a couple 100 million years. They're actually that a lot of that diversification was post-Cambrian. So you get that actually more recently than a lot of animals.
Oh, yeah. So you got. It's been an hour, so you gotta go because of your baby's fever.
I should be responsible and get myself to bed at a reasonable hour this evening. So I think what I'll leave everybody with is endogenous retroviruses, super awesome, make up 8% of the human genome. And the really cool thing for evolution is you can follow the phylogenetic history of vertebrates just based on endogenous retroviruses. And you can reconstruct all the relationships just based on herbs, which is so cool.
Yep. Another layer to the puzzle.
Yeah.
Morphology, DNA, and now viruses.
Definitely a lot of fun. And that's why I'm studying biology.
The best subject.
Yep. I love it.
Before we go, you want to shout out your show if you're on your show tomorrow or not? You want to shout it out?
Sure. So yeah, subscribe to my channel, subscribe to Dr. Dan, subscribe to this channel here, and I am planning a debate coming up soon with Gavin on the subject of the resurrection of Jesus. That will not be on Standing for Truth's channel. It will probably be on Praise's channel, but stay tuned. But it will definitely not be on Standing for Truth's channel.
Yeah.
So stay tuned.
Yeah, we're having a little issue.
I don't have anything coming up in the immediate future. I might have some.
Oh yes,
I might do a stream later this week or next week. But then the really exciting thing is March 22nd. I am having Michael Behe on to talk about irreducible complexity. That's going to be super exciting. That is March 22nd on my channel, Creation Myths, at 1 p.m. Eastern. Michael Behe is going to talk to me about stuff.
Yeah.
And
one thing that I am working on is a new series called Yuckfield. So basically, it's just a bunch of the funniest clips from young Earth creationists and poking some fun at them.
Okay, so I can have your. Oh, nice.
If you want, I'll give you a backstage.
Oh, awesome. So you're not going to have your usual Wednesday show tomorrow then, your baby because of the fever and stuff.
Oh, I don't really have a normal Wednesday show. I just occasional. I don't know. I don't have anything planned for tomorrow. We'll see. Maybe if I do something, it'll be short notice. I don't have anything planned for this week though.
If you have,
I wish I had a regular schedule, but I don't.
Yeah, if you have time tomorrow, I would love to have you come on to talk about your PhD thesis and other stuff.
Cool. Yeah,
I will definitely do that at some point.
Yeah.
I don't know about tomorrow, but I will definitely do that because I will tell you. You ask anybody, what did you do research on, or what did you do research on? Oh, they will just go for a long time.
Oh yeah.
Get ready to talk about cytosine, my favorite thing.
Yeah. Well, join me this Saturday. I have no time yet with me, RJ, and our friend Eric. We're talking about the drama of the drama. People an uprising. People are creating it over the Dr. Seuss books.
Oh goodness gracious! Oh, well, that'll be a fun one.
Yeah.
Right. Until then, check. I'll have the link to their channels in the description below. But in the meantime, never stop learning and enjoy the randomness. I'll see y'all next time. Bye.
Bye-bye, everyone.